Uh, Good day. Good day, all of you. Today I'm going to be teaching about proteins, bio 111 lecture 14 proteins. We're supposed to have a lesson in the morning, but we are unable to have a lesson. So at first we say that there are four organic molecules, which are carbohydrates, proteins, lipids, and nucleic acids. We say that these are the building blocks of lives. Uh, in this lecture of proteins, in this lecture of proteins, we are to study um, okay. As you can see, the representation of a human body. Um, Inside the human body, like there is inside the human body, water takes up 70%. The macromolecules takes up this percentage, and the ions and molecules takes up this percentage. From these macromolecules, which we are studying, we can see that proteins they take up the larger portion. So in other words, we can say uh, that the relative proportion of the macro molecules in a human body, uh, you will see that proteins they occupies a lot of space as compared to other macromolecules. So proteins are large. Proteins are large. They are complex molecules. Complex are large and complex molecules. Uh, we can say that they are made up of monomers, uh, a smaller unit called the amino acids. If you can reflect back to carbohydrates, you said that carbohydrates are made up of saccharides, and proteins are made up of amino acids, which are attached to one another in chains. The same principle is going to continue applying, that if you have a amino acid here linking to another amino acid, linking to another amino acid, you are going to form a protein. The amino acids are basic building blocks of proteins, or theirs are the monomers of proteins. Same applies to the saccharides, which are monomers of the carbohydrates, which are monomers of the carbohydrate. The proteins are required for the structure, function, and regulation of body tissues and organs. You might, if you might remember this, you guys were learning in high school that proteins are being needed to repair tissues for growth, actually, because growth starts by repairing of tissues and also the replications of cells. Most most proteins are functional, and they have a diverse group of biomolecules. The functions of proteins are involved in a lot of things. Proteins they can function as enzymes, which we call the control molecules. Proteins can function as the structure in keratin and collagen. Proteins can function as the carrier and for transportation, such as the glycoproteins. Proteins can function as the receptors and the binding sites for the defenses. Also, protein, they can function as contraction structures. When you now look at the myelin and the actin, the muscles. So the functions of proteins are so diverse because they, as you see, they are major in the body. There are a lot of proteins in the body. They are ranging from enzymes and from muscles, ranging from carrier proteins. They are also ranging from what? From receptors and binding sites. Hormones are also proteins. It means we can say that the other function of a protein is to signal. And most proteins we see them in what? In storage. Proteins are in bean seeds. When two amino acids link together, they form what you call a dipeptide. We can say a peptide is an amino acid. Is an amino acid. So when three amino acids link, they form a tripeptide. They form a tripeptide. 4 to 10 amino acids they link, they form an oligopeptide. 4 to 10 amino acids linking, they form an oligopeptide. More than 10 amino acids, when they link, they form what you call a polypeptide. They form what you call a polypeptide. Proteins in the body and diet are long polypeptides. It means the proteins which we normally find in the body or which makes up the body sometimes they are polysaccharides. You know about the process of digestion. All of these, they are going to take place in direction, but the proteins that you are going to be seeing most of the time are the polypeptide. Polypeptides are long chains of amino acids. We can say that these proteins can have one or more peptide chains, which sometimes can be folded or bonded together. Proteins have a complex 3D shape. The chemical structures of proteins. Proteins, they exist in 4 structures, which are the primary structure, the secondary structure, the tertiary structure, and also the quaternary structure. So the primary structure of proteins is a sequence of amino acids. Whenever they're still uh, in a single peptide chain, or can we say a strand, we can say that they're existing in a primary structure. So the sequence of amino acids is determined by the DNA. So in this sequence of the primary structure, a slight change that can happen in the sequence can affect the structure and the function. So this sequence of the primary structure is going to determine the what? The sequence of the second of the secondary structure, because uh, they are go there's a way in which they link. There's a way that they they link as a result affecting their folding. You can see it. You can see the example of a primary structure there. So we can say that the proteins have what you call an amino group there, and also what you call the carboxylic group. This is now the amino acids, bo lysine, bo glycine, bo alanine. So you see that there are 20 amino acids, and most of them you find them in primary structure. And there is a way there is a way that they link, meaning that whatever they link, or however they link, is going to affect the secondary structure. You can see also the structure of any primary structure of proteins. So at this level, you are not supposed to know zonete, glycine, valine, and also cysteine. Ngitshela motho rona ya tshwanetse go tla le focus mo go itse gore a protein in a carboxylic group le amino group. But in understanding test, this structure are going to claim it. The first structure there is a primary structure, this one. Secondary, tertiary, and quaternary. You can see that the process or the complexity of the protein increases going downward the structures. So in the primary structure, you can say that proteins will be assembly. In the secondary structure, you say that proteins will be what? Will be folding. In the tertiary structure, you say that proteins will be packing. In the quaternary structure, you say proteins will be what? Interactions of what? Of the proteins. Let's study the structure of an amino acid. An amino acid, this is where now the topic gets interesting. In amino acid, we have a central carbon. This is a central carbon. And after a central carbon, then amino acid is going to have an amino group. This is an amino group. After that, the protein, the protein will have a carboxylic group. This 1, 2, 3, and also this hydrogen, they are always there at the in the protein. For you to tell me that this molecule, this micromolecule is a protein, you need to know, or you need to be able to identify the amino acids, the central carbon, the carboxylic acid, and also the H atom. This is a hydrocarbon chain. Sometimes can have rings here and all these, but is an R group or a side chain of a protein. So these are the functional groups which they need to be there for you to classify something as protein. So there are variable group from 1 to 20, the R groups. As we know that these are going to be now amino acids attaching to each other. As we say that there are 20 amino acids which can form a sequence in this to make your name the R group or the side chain. So let us look at an example of some specific amino acids, and you need to be able to name them. That okay, this is a generic amino acid. This is a resemblance of what an amino acid should have. Should have one of the 20 unique side groups, an amino group, and a H, a central carbon, and also a carboxylic group. So glycine is the simplest amino acid that is there, and most of the time we'll be drawing glycine. You need to be in a position to know how to draw glycine. You need to be in a position to know how to draw an phenylalanine. Phenyl means this is a sort of a phenol there, which I mean sorry, a ring. So that's what we call it, is a phenyl, a alanine. So building blocks of proteins, we say that whenever you build the protein, an amino acid and an amino acid, they have to come together to form a polypeptide chain. So the reaction there you see is a dehydration synthesis. Why do you call this a dehydration synthesis? Because we see that water is lost. Water is lost from the during the reaction. Uh, So, and the bond which is going to be formed now, this bond in red, when an amino acid comes to reacts with an amino acid, is called the peptide bond. Uh, the linking amino group or in one amino acid to the carboxylic group or to of another amino acid, there is a linking there. So you can see that this bond, the CN bond, has been formed, and we call it a peptide bond. We can say that a polypeptide will have the N terminal, which the N terminal is going to be called the amino end, and also the carboxylic end, which is a C terminus. C terminus. The repeated sequence of this bond, the NCC of the polypeptide, is called the backbone of the polypeptide. So you guys need to go and study further on this. So this was supposed to be our lesson, which I missed. I hope you guys were ready. You just I use this as a reference, and I think you now appreciate the work faster.